So good morning once again. So today we are going to build on from what we looked at. So this is once again teacher Dr. MUGOMBA Ronald. We are going to build on what we looked at previously. We looked at mitosis previously, and we looked at the mitosis and the cell cycle. So today we are going to build on that. So in meiosis, as we are going to see, meiosis involves a number of items that we shall look at. Usually, we usually have one cell dividing into 4 cells, and this kind of meiosis usually takes place in the type of cells known as gonads. The gonads are the cells which participate in meiosis. Originally, in mitosis, we said mitosis is a type of cell division whereby we see cells dividing into 2 identical cells, diploid cells. So, in this case here, we are having one cell dividing into 4 cells, but each of these 4 cells are having half the number of chromosomes as their parent cells. It happens only in the gonads. The other one, mitosis, happens in the cells known as the body cells, other cells, somatic cells other than the gonads. The gonads can be the testes and the ovaries for the case of animals. And then when you look at so in plants, it usually takes place in the ovaries or the anthers. So so you can see here, just showing an image that I placed here that tries to show you what is meant, how fertilization takes place. So when you look at now, we need to understand some concepts here before we can go ahead and look at the different things entailed in meiosis, and that is, we need to understand the meaning of what they call homologous chromosomes. Remember, yesterday we looked at the cell cycle, and we said that cells usually undergo before division happens, the cells prepare by duplicating genetic materials. So homologous chromosomes are usually a pair of homologous chromosomes. There is an event that is so specific to meiosis, and that is homologous pairing, or what they usually call synapsis. It is specific to only only meiosis. So a pair of chromosomes which bear almost similar genes is known as homologous chromosomes. They're usually pairs. They exist in pairs. Pairing. Chromosomes come and pair up, and then they code for somewhat similar or unique similar characteristics. So in human beings, we have about 23 pairs of homologous chromosomes, and that makes it a total of 46 chromosomes. Houseflies have 6 pairs, chicken have 39, apples have 17, dogs have 29, cats have 19. So that whole outline, a printout of all the chromosomes that you have in your cells, is known as a karyotype. A karyotype is a printout. It's that combination of all the chromosomes that are possessed by a cell. That is what we call a karyotype. So an image of an organism's genetic makeup. So so this one here that we are having here is a normal man or a normal male, simply because when you look at his chromosomes, you realize that he has a Y chromosome within his karyotype. So having looked at that, so you can clearly see that what a homologous chromosome looks like. So that is how a homologous chromosome looks like the gene for eye color. We know that the gene for eye color, and you can see that within the chromosome, on the chromosome, there are a number of things that we usually talk about. We usually say that these genes on the chromosomes usually occupy specific locations within the chromosome, and those locations where we get certain genes on the chromosome are known as gene loci. A gene loci or loci, there are two. Loci is plural and locus is singular. So gene loci are usually those places where you find different genes. Like the gene for eye color, you realize that the loci of gene for eye color is usually present on the chromosome. Now when you look at those chromosomes, they appear in pairs simply because it is an added advantage in a way that in case a mutation happens in one of the genes, most of the mutations that you will encounter that are lethal, most of them are usually recessive mutations. That is to say, for them to cause their adverse effect to a human being, we need to have the recessive gene being homologous. What do I mean when I say the recessive gene must be homologous? That means that both chromosomes in the pair should have the recessive gene, the gene for the mutation, which is a dangerous one. But since now you have two, in case one is affected and it is carrying the defective gene, it is compensated for. This other gene on the other normal gene can compensate for the and cover up, it masks the faulty gene. So you can see those so you can see usually these homologous chromosomes, we inherit one of the component of the pair from our parents, and the other one from our mother and from our dad. So usually we have the paternal one and the maternal one, but all of them are carrying genes for similar characteristics. That is to say, let's look at maybe genes for eye color. You will inherit some components from your mother, and you'll also inherit some components from your dad. Let's say gene for hair color. You will inherit a component from your mother, and you inherit a component from your dad. So usually, as yesterday I told you that in cell division we do not talk about the preparation phase. The preparation phase, known as interphase, is just a component of the cell cycle, but it does not take part in the division. So when we are looking at division, we usually now start with the prophase. Now here, in meiosis, it involves two cell divisions, or two nuclear divisions, as we shall see. It involves two nuclear divisions, and we are going to start with the first nuclear division. So when you look at the first nuclear division that we are having here, so it will be having prophase 1, prophase 2, it will have metaphase 1, metaphase 2, it will have anaphase 1 and anaphase 2. It will have telophase 1 and telophase 2. Nuclear divisions are usually 2. So we are going to begin by looking at prophase 1, the events which take place. Usually, prophase 1 is the longest. As we are going to see, it's the longest simply because of the very many different things that happen within prophase 1. We are going to see them, the different things that usually happen during prophase 1. Then it is the longest. So usually, as we saw in mitosis, the chromosomes, they condense from their thread-like characteristics. They were once thread-like characteristics during the preparation known as interphase. Now here they condense into visible chromosomes, which are made up of a pair of chromatids. And then the other thing that we need to note, what happens is what we usually call synapsis. Synapsis is a process by when the chromosomes come together, a pair of homologous chromosomes come together. That is synapsis. That is to say, what the other chromosome from your dad and the other chromosome from your mom, they come together, they form a pair. That is homologous chromosomes. So it is known as a triad. The reason as to why it is known as a triad, you can see these ones, there are two. It's known as a triad simply because sorry, it is known as a tetrad simply because it is made up of four chromatids. That is why they call it a tetrad. A tetrad simply because it is made up of 4 chromatids. That is why they call it a tetrad. So 2 from the maternal side and 2 from the paternal side. So so you can see now they have come together, and now they have synapsed together. They have formed what we call a bivalent. So this component, this structure that is formed during synapsis is known as a bivalent, or sometimes it is known as a tetrad. So non-sister chromatids. So usually what happens, usually as we are seeing, you can see the chromatids are now non-sister chromatids. In the triad, we have non-sister chromatids. They are called non-sister chromatids simply because they are coming from different chromosomes. These chromatids, which make up one chromosome, are called sister chromatids. The non-sister chromatids are the chromatids that are coming from different chromosomes. So that's why they're calling them non-sister. One from the paternal, one from the maternal side of it. They are called non-sister chromatids. Okay, so these are the sister chromatids are the ones which are making up one chromosome. The triad is made up of 4 non-sister chromatids. So the tetrad is made up of 4 non-sister chromatids. So, so another event after synapsis is done, I have said synapsis is the pairing. Chromosomes pair up. They randomly pair up with each other. And then next step after they are paired up is what we call crossing over. Crossing over usually takes place in the tetrad between the non-sister chromatids at the ends. What happens is these chromatids usually they break and rejoin. As you can see clearly here, after synapsis is done, usually pairing of the homologous chromosomes takes place. So when the homologous chromosomes have paired very well, what happens next is we usually see, you can see this area here. This area here, you can see clearly that now at the end of each of the chromosomes, we see some breaking. So the end of the chromosome breaks. One chromosome, the maternal chromosome, its non-sister one of its chromatids breaks, and then the maternal one also breaks, and then we see exchange. It breaks and then exchanges. A piece from the maternal is transferred to the paternal, and a piece from paternal is transferred to the maternal. That phenomenon is known as what we call crossing over. And this crossing over as a process is very important when it comes to bringing about the differences that we usually observe among members of the same species, among even sisters. Have you ever been there and you wondered why your sister is relatively different from you, yet you were produced from the same father and mother? Even your brothers, you realize that your brothers will look a little bit different from you. It is simply because of this phenomena which is known as crossing over. So during crossing over, we usually see exchange of genetic materials. Exchange of genetic materials which happens in the non-sister chromatids. Why is it non-sister chromatids? Simply because the non-sister chromatids are coming from different chromosomes, one from the paternal and one from the maternal side. So they usually break and then rejoin, then break and rejoin. What happens is as they break and rejoin, we see genetic mixing up. So we come up with new gene combination as a result of crossing over. So what happens, the role of crossing over is that it creates new gene combinations. These new gene combinations that are usually created are the ones which bring about the basis of the differences that exist among individuals of the same species. Even individuals who are from the same parents. This is the phenomenon known as crossing over. It is very, very, very fundamental in bringing about variations, and it is variations that drive the entire process known as natural selection, as we shall see in the later chapters. So having looked at crossing over, so I've talked about crossing over, bringing about variations, provide variations. That point where we usually see the non-sister chromatids joining, the breaking and then rejoining, that point is usually known as the chiasmata. You will find some books calling it chiasma. It's the same thing. Chiasmata, chiasma, all the same, site of crossing over. So we have seen that, you've seen that already. So these are variations. You can clearly see that here, this is a new gene combination that has been formed. You can all notice with me that this is a real new gene combination that has been formed. Completely new. New. Originally you had this one here. You had these two, which joined at the synapse, they joined at the chiasmata. Crossing over took place, and after crossing over took place, we formed new chromosomes because they separate thereafter. Now, having looked at that, so this is a variation. So you can clearly even see it from here. These chromosomes, you can see that now crossing over has taken place between the homologous chromosomes. You can clearly see here. You can also see here. This is all crossing over. So now the next thing after having, so those are, that explains why it is very long stage. It is very long. It takes 90% of the time. simply because of those events, and those are very important events. I've talked about crossing over, which happens after synapsis. Now, the next step that we are looking at is metaphase 1. In metaphase 1, you can see that the chromosomes align themselves. It is the shortest stage in meiosis 1. It's the shortest. Usually, we see homologous chromosomes pairing up, and then they align themselves at the spindle equator, like in mitosis. But as we are going to see, there is a very clear difference between between metaphase 1 and metaphase of mitosis. Simply because when you look at these chromosomes which have aligned themselves at the spindle equator, you can clearly see that they have organized themselves in a way that they are in pairs. So that alignment of chromosomes at the spindle equator in pairs, you can clearly see that they're in pairs. That means that, like, it is not like previously in mitosis where we saw that the chromosomes arrange themselves independently of each other. These ones arrange themselves in pairs. The other side, in mitosis, we saw that chromosomes were aligning themselves independent of each other, each of them separately. And now we are seeing here, these ones here, the difference is in metaphase 1, they are organizing themselves in the at the spindle equator. They are organizing themselves in pairs or homologous pairs. Now, when you look at it clearly here, you can see that very well. Independent assortment takes place. You can see independent assortment is taking place, and this is another source of variations, as we are going to see. So we usually see pairs of homologous chromosomes homologous chromosomes lining up independently of other pairs, orienting themselves towards the poles. And it is random. Being random, that makes it a source of variations, as we shall note. So we have already noted that crossing over is one of the sources of variations. The second source of variation exists in independent assortment. Independent assortment is, I'm asking myself a question. When you look at these chromosomes, when they are aligning themselves at the spindle equator in pairs, they do it independently. The one that comes to the left, let's say we have two poles. We have the pole at the this left side, and then the pole at the right side. The one that comes to the left does not first ask the one that is going to the right. It is independent. So you can have the maternal on the right. When you look at, let's focus on maybe the chromosomes which are responsible for hair color. Okay, so for hair color, let's say the one that is going to the right is going to be for the one that came from the mother. Then the one which goes to the left, that came from the father. Then when you look at maybe for the ones for maybe, let's say, length of hair, let's say, or eye color. Now the one for eye color, it might be opposite. The one from the mother might go to the left, and the one for the mother might go to the right. This explains why sometimes you can find a boy having characteristics like his mother. Probably, or maybe the hair is like the one of his mother, simply because maybe the gene that is, when they were organizing themselves at the spindle equator, this boy maybe probably came from the cell which obtained the maternal chromosome. This boy was produced from the cell that came from the maternal chromosome. I'm just assuming. You will find some boys having characteristics like that, or they have more of the characteristics, maybe in terms of eye color, they look more like their mothers, simply because of this independent assortment. When they are aligning themselves at the spindle equator, it is known that all the paternal chromosomes go to the left and the maternal go to the right. That is not happening like that. They organize themselves in an independent way. They reshuffle themselves. The one for maternal for hair color might be on the left, and the one for maternal for maybe eye color might be on the right. So that is how they assort themselves, and that is a source of variations in nature. And I've explained it very well now. So that is metaphase 1. So metaphase 1, even in metaphase 1, we see some variations coming in here. We looked at prophase 1 and we saw some variation coming in here. In metaphase 1, we're also seeing a variation coming in. Why is it coming in? Simply because of the way they organize themselves at the spindle equator. The ones that are going to the cell that will be on the right will be different from the ones that are going to the left. It's not that all the paternal go to the right and the maternal go to the left. No. You can have some paternal going to the right, some maternal going to the left. Maybe the one for eye color for maternal goes to the right, and then the one for skin color of maternal goes to the left. So, that intermingling, but the major thing that we need to note is that they organize themselves at the spindle equator in pairs. Good. So we can then thereafter go ahead and look at the next phase known as anaphase 1. Now, in anaphase 1, as you can see in the picture, clearly you can see that these chromosomes now are going, the tetrad is going to separate. Remember, the tetrad was originally, they were originally all together. Like even in metaphase, mind you, in metaphase they are still together in a pair. But now in anaphase, ana means away. I remember I told you ana means away. So when you're looking at anaphase, it is moving away. So this pair starts to separate. In mitosis, we saw it was the chromosome that was separating. The chromatids were the ones separating. But in anaphase 1 or meiosis, we are seeing the homologous chromosomes are the ones which separate. So you can see that is a very unique difference. Here we are taking the whole chromosome with the two chromatids. The other side we are only taking a single chromatid from one chromosome. So you can clearly see now the spindle fibers have now formed already, and now they are dragging. You can see the proteins within the spindle fibers start to contract, and now they pull the homologous pairs apart, and they separate them towards the opposite poles. So a point to note is the fact that when you look at it clearly here, you can see the sister chromatids remain attached to each other. You can all note with me that the sister chromatids have remained attached to each other. What has separated is the bivalent, or the tetrad has been separated. So you can see now we can go ahead and now look at telophase. So I'm leaving out these things, like for instance the nuclear envelope reappears, those things of the nucleus reappears, the centrioles go to opposite poles. Now those ones I'm leaving them out because we talked about them in details. Here I'm highlighting clearly the different components that are specific to meiosis. So each pole is now having haploid set of chromosomes. So we have the other side, we have, but however it is still doubled. Remember we started with 2n. So what happens is, since we have 2n, what usually happens is during the proliferation stage we multiply that 2n to 4n. So when we go to 4n, from 2n to 4n, now at this stage we have just made diploid we have now separated the 4n. Now we have 2n on one side, 2n on the other side. So now that is what we call telophase 1. Now in telophase 1, sometimes what happens, cytokinesis follows thereafter. Sometimes cytokinesis does not follow thereafter. But now this one here, after the chromosomes have reached the opposite poles during telophase 1, then the cytoplasm can separate. Now after, what happens thereafter is meiosis 2. Now the cells that were now originally they were the cells which were formed during meiosis 1, now they are going to undergo division to form, each cell is going to divide again to form other 2 cells, and that gives us a total of 4 cells. But during meiosis 2, it should be important to note that the cell does not go back into interphase. There is no interphase. And that explains why now we are going to see halving of the genetic material. If we were to go to interphase, we would not have the genetic material. But since we are not going back to interphase, we are not going to have the genetic, we are going to have the genetic material. So there is no more lubrication of the DNA. And now, another note that I can say is meiosis 2 is similar to mitosis, as we had outlined earlier. So you can clearly see that prophase, you can clearly see the prophase, during prophase, they start, the chromosomes condense, then metaphase, they align themselves at the mesophase plate. Then after metaphase is done, the chromatids, now the sister chromatids are pulled apart from each other. They separate at the centromere. During anaphase 1, we saw them separating from the bivalent at the point known as the chiasmata. Now here it is the centromere that is going to be pulled apart. And then they go to the opposite poles. That is anaphase 1, anaphase 2. So now you can see that now this is telophase 2. Now where you can see that each cell is having just one chromosome. I told you that by the time the cells start migrating to the opposite, by the time the chromatids are migrating to the opposite poles as they are dragged by the spindle fibers, they're no longer called chromatids. Now they are fully fledged chromosomes. Now you can see that during telophase 2, you can see clearly that it is the same as that in mitosis. The nuclear envelope reappears. You can see that now the nucleolus also reappears. The nucleoform, you can clearly see that the chromosomes decondense and they become more thread-like. So that now in preparation for the next stage, now we can see cytokinesis taking place once again. And now we have our 4 daughter cells being produced, and the chromosomes, now we can now say that these are now chromosomes and they are in their single condition. Then during the next stage, they will go ahead and duplicate and they become 2-2. So you can see it usually happens in the gametes, that is saying the sperms, the eggs, and the ovules, the pollen grain. So this is still an illustration that shows you how the whole process takes place. Prophase 1, metaphase 1, anaphase 1, telophase 1. So you can see all those events that we have been talking about, each of them. You can see all of them. You can see all these stages. Now this is meiosis 2. You can clearly see that during meiosis 2, you can see the chromosome. Now it is the chromatids that were pulled to the opposite poles in prophase 2. Sorry, in meiosis 2, we can see that when it comes to anaphase, it is the chromatids that are pulled to the opposite poles. So we see separation of the chromatids at the centromere. You can clearly see that. So it is very important for gamete formation in males and in females. And that is what we usually talk about in gametogenesis. In gametogenesis, we usually have the sperm and the ovum. So we usually have what we call oogenesis and spermatogenesis. So they are usually involved in spermatogenesis and oogenesis. So I've been looking at a summary of the different events that usually take place during the process of meiosis, and we have highlighted in our discussion, we have highlighted the fact that during meiosis